I think greatly from the data processing side of things, AI would be really helpful because you get a lot of data. At the end of the day, that's also another bottleneck is how do I reduce my data to meaningful results, like conclusions at the end of the day.
Welcome to Data and Biotech, a podcast from Cordi where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
Eshani Galermo, welcome to the Data and Biotech podcast.
Oh, thank you for having me.
Awesome. Well, just to kick us off, would you mind giving us an introduction to you and your background and what brings you to AAPS Pharmacy 360?
Hi, I'm Eshani Galermo. I hold a PhD in analytical chemistry from UC Davis. So I studied mass spectrometry there. And then I worked for a CRO working on pharma and biopharma workflows there. And then I shifted to SCIEX, and I've been with SCIEX for 5 years now. And currently I'm the staff scientist there. Still working on pharma and biopharma workflows. So
what drew you to work in, bioanalysis and in bioanalysis equipment?
Yeah. So, I started off at a CRO, right, out of grad school, and we were working on a lot of bioanalytical workflows there, and that got me really excited. That was my first hands-on experience with a lot of different customers in biotech and pharma, biopharma, so it was very exciting. And then, I come from a mass spec background, so eventually, I guess it drew me to a mass spec vendor, and I ended up with SCIEX.
Starting with sort of like the customer side, can you give us some insight into, what are the types of questions that biotech companies are trying to answer using quantitative bioanalysis that comes like off of mass spec machines?
Absolutely. So one of the main things is how can I be able to detect the compounds in my sample, right? And what levels? And can I reach the levels I'm trying to reach? So that's where sensitivity comes into play. And so a lot of the instruments you will start to see, there's a lot of innovation, a lot of features going in to be able to cater to that. And as well as, specifically for pharma and biopharma, you have these samples in different types of matrices, biological matrices. So they're in tissues, they're in liver samples, plasma, serum. All of that is very complex, and being able to pick out one particular target analyte from that matrix is very challenging. So that's one of the critical factors. So going into sensitivity is number one. Number two is they're trying to see if they're able to measure across a wide range of concentrations. So that's where, you can have samples with lower abundant compounds, and then you can have samples with higher abundant compounds. And can I be able to measure all across the wide range? So that's another. So that would be their second question. And then also, there's characterization. Can I figure out what the structure looks like? Where is the modification happening? Can I figure out if those modifications is going to cause, different types of effects down the road? So as they're going through drug development, that's a big question as well. So those are all the types of questions we're typically getting from customers as a mass spec vendor and trying to find solutions for them.
So I'm interested in, what is it about the traditional mass spec that, doesn't where does it fall short, or where does it not quite get bioanalysis companies or, teams to the place of sensitivity that they really need?
Yeah, we have different types of mass spectrometers that are out there. But if I had to couple it or categorize it, it'd be like nominal mass spectrometers and high resolution mass spectrometers. Traditionally, speaking of bioanalysis, since we're at AAPS, right, a lot of that quantitation workflows have been on nominal mass spectrometers. That has been the gold standard. But nowadays, we're starting to see a lot of analytes being, more complicated, more complex in structure. And then once again, you have those types of analytes in very complex matrices, biological matrices. So being able to detect those types of structures as well as quantify them becomes a very big challenge. And so there's been a huge push for high resolution mass spectrometers in the space of bioanalysis to be able to cater to those challenges. And so those specific unique workflows are now pushed on to the high resolution mass spectrometers.
Yeah, and if I'm understanding correctly, like some of the new molecules that are coming about, like GLP-1s or like peptides, like these are sort of lend themselves better to high resolution, or those are some of the complex molecules that you're talking about.
Yeah, I mean GLP-1s, but also like the oligo field, right? So you have oligonucleotides, you have siRNAs, you have antibody oligoconjugates. So you got, antibody part to it, a linker, and then an oligo. So there is a lot of, these structures that are coming out with additional components that you're needing to be able to either verify or identify or quantify. So all those aspects need to be catered to. And a lot of times you often face some type of, complex issue when it comes to the analytical side of things, whether it be like sensitivity or selectivity, being able to pick up your target from a background, or you are trying to quantify a specific structure that's very hard to do so on mass spec sometimes. So
that makes a lot of sense. So, my understanding is that the technology has evolved for high resolution accurate mass has evolved pretty dramatically, and that these tools historically have been more qualitative discovery tools rather than quantitative discovery tools. Can you maybe characterize the difference between like qualitative discovery and quantitative discovery in mass spec, and then maybe explain like how the technology is enabling high resolution accurate mass to do both, as I understand it?
Yeah. From the perspective of what we've been developing in-house, qualitative, right? If you were to figure out or differentiate between qualitative and quantitative is more looking at how can I structurally elucidate my compound, right? Can I figure out what are the components that are present or the modifications? What does my compound exactly look like? For quantitative workflows, it's more looking at the actual amount or concentration levels of my sample, right? Or my target compound in my sample. So traditionally, like you said, high resolution has been pushed for more qualitative workflows. But nowadays, because with a high resolution mass spectrometer, what you can do is be able to differentiate your target analytes from your background analytes or ions better. And so one of the key advantages is that selectivity based on the high resolution that you are getting from that system. And typically that's advantageous in the quantitative field because that helps you be able to reach a lower level of quantitation, right? You can be able to get to that lower concentration levels better, right? And so because of these kinds of advantages in the high resolution mass spectrometer, people are all looking at high resolution. And it's also flexible because you could do both qual and quant. And so because of that, a lot of people are starting to look into high res and, trying to incorporate it into their daily bioanalytical work.
My understanding is there are like a bunch of other, like in addition to being able to being more selective and more sensitive, there are some other benefits to using an instrument like the XenoTOF 8600. So, for example, the idea that you can use less sample, that you don't require as much sample in order to be able to detect what's happening with your compound, but also that like there's this opportunity for your workflow to be speedier. Can you maybe verify it, if I'm understanding correctly, that those are some of the benefits they bring to the table? And if so, like maybe explain like how using a tool like the XenoTOF 8600 like allows for you to use less sample and or get your workflow done faster.
Yeah, so the XenoTOF system, we have the 76 as well as the 86, right? So the XenoTOF series in general, we have a specific area in the system where we introduce a trap. And basically what it does is, your analytes are injected into the system, they turn into ions, and then the ions flow in, and the user selects which particular ions they want to look at. Then it goes under a cell where it fragments into pieces. Then it goes into the time of flight, which is our mass analyzer.
Right.
And typically, when the fragment ions going to the time of flight, there is a ion transmission loss. And because of that, we put in that trap to kind of bundle it all together, and they send it into the mass analyzer and you detect it that way.
Right.
And so from that, then you are able to get the best sensitivities when it comes to a QTOF mass spectrometer, because traditional QTOFs don't do that. And that allows you to make sure that you're not losing that sensitivity of what you injected initially because of that, just the intrinsic nature of QTOFs, right? Because of that XenoTrap, the XenoTOF series has really pushed the boundaries for sensitivity. And you'll see that, both with the 76 and 86, that really allows you to capture even, the lower end as well as the higher end.
That was really interesting to me. And also this idea that like there's this duty cycle that mass spectrometers
essentially it.
have, where typically in a mass spectrometer, if I'm understanding correctly, the ions are being sent into the time of flight detector, but the time of flight detector is only on for a very short period of time, and so there's this idea that the XenoTrap is sort of like holding the ions and only sending them when the detector is ready to actually detect, and so you're able to get that signal much more strongly. Am I thinking about that right, or understanding it right?
So it's a pulsar essentially, so you have to bundle it in order to send that packet of ions. Otherwise, you get a continuous flow, you'll definitely be losing ion transmission, right?
Find that aspect of this really interesting. So now that we have a little bit of background on that, on mass spectrometers generally, can you just outline typically what is the data that comes off of a mass spec machine, and like how you analyze it in order to draw get some of the answers to some of the questions that you discussed earlier?
Yeah, absolutely. So let's say for a quant experiment, right? If you're trying to calculate the concentration levels that you injected originally into the mass spec, what you would do is at the end of the analysis, you will get a certain intensity as well as the M/Z, or the mass to charge ratio of your monitored fragment ion. So in that case, you would take that, it would come in the shape of a peak, right? So you would take that peak, you would integrate it, use the peak area, and then back calculate what that concentration would be.
Right. So there was this calibration that happened prior to it, where you sort of understand the relationship, as I understand it, between the area of the peak and like how that maps to what the concentration of the compound is.
Yeah. So essentially you will get like the retention time, and then you would get the intensity, and you would get several different peaks according to whatever you were trying to measure.
Right.
And each of those peaks also has information of what particular precursor ion or the original M/Z of your analyte, as well as the fragment ion information.
Yeah. So you can distinguish all of the things that are present, and then how much of each thing is present, and then the conclusions that you draw from that sort of depend on the nature of the question that you're asking. But the ability to detect sort of small variations in the things that you're detecting, yet then also detect exactly how much is present. Both of those things are sort of elements of the high resolution accurate mass. Am I thinking about that right? That's awesome. So, you have, so if I understand correctly, the 8600, that this newer piece of equipment, offers this, 5x to 20x gain in sensitivity. From a bioanalysis perspective, like what does that get you in terms of your workflow, or in terms of like your ability to, get answers faster, less expensively, or, to questions that you wouldn't have even been able to ask previously?
During drug development, you have different levels of the drug development process. And typically you would have your particular therapeutic in different types of samples. It could be in mouse samples, like in your preclinical studies, or clinical studies would be in human samples. And a lot of times you would try to minimize the amount of the sample volume, right? Just because of the nature of it's an invasive nature, right? So in that case, you would be, having small sample volumes that then have a smaller concentration of your compound, because over time you're monitoring your drug over time, and as well as metabolites, which is basically your body metabolizing the drug and turning it into other different types of other structures. And so as the time goes on, the concentration drops, and you can imagine small volume sample, less concentration of drug, therapeutic. How can I be able to detect that? And to the lowest level, so you can safely say, this is the tox level, this is the safety, this is how, this is efficacy, and then I can push this drug out safely, right, to the public. So it's really important to be able to have an instrument or a platform that can be able to target those low level concentrations, especially in those preclinical and clinical stages.
Right, and I can imagine from a regulatory perspective, depending on the nature of the compound that you're bringing to market or the therapeutic that you're bringing to market, like you might have obligations to reach these like very lower levels of quantitation that you might not be able to reach with your typical triple quad mass spec machine. Am I thinking about that right? Or
oh yeah, there can be cases in terms of the compounds that have those, like I mentioned earlier, with the background issues, or you need better selectivity. You're able to do that easily on a high resolution mass spectrometer and be able to, safely push out the drug.
So we've talked about LLOQ, but I'm interested in one of the things that you talked about in your presentation last night with this it was this idea of sort of a broad linear dynamic range. So I'm wondering if you could like define what a linear dynamic range is in the context of what we're talking about, and then maybe talk about why that's like critical for streamlining late stage clinical studies and making sure that the data integrity is there.
Yeah, as I was mentioning earlier, right, when you're looking at the drug over time, right? If you're dosing, it will go from high concentration to low concentrations, and it'll give you a range. And basically, being able to measure across the lowest range and to the highest range is very critical. And the XenoTOF 8600 system has that new optical detector that allows you to be able to capture that wide range of concentrations. And you saw, right, with different examples, we can reach up to 5 orders of magnitude in some cases. So it's really critical because, you can have samples that can have very high levels of your analyte, and there can be samples with very low levels. How can I have, a full assay that can be able to measure that?
One of the things that came up as I was researching the 8600 was also this concept that by having this much greater sensitivity, leveraging the full scope of the sample that you're pushing through, you're getting so much more data that allows you to ask more follow-up questions about like sort of the composition of what's in your sample. First of all, like, is that understanding correct? And if so, like, are there examples of questions that people ask like after doing mass spec that the XenoTOF 8600 like enables them to ask?
Absolutely. There's times when you can open up the instrument and just get the full profile. Basically, hey, I'm injecting the sample that can have multiple analytes. What do I have? And then sometimes you'll see peaks there that you don't know any information about. And so those are flagged. And because the system also allows you to collect fragment ions, which is pieces of the initial ion, right? You could put the puzzle pieces together essentially and just be able to elucidate what that structure would be like. There's libraries available, and sure, like a lot of companies have in-house libraries as well, and they can be able to figure out what that structure would be like.
Having that higher degree of sensitivity means that you're going to be able to pick up on those things that wouldn't the smaller things that wouldn't otherwise be there if you had like the 7600, for example. Okay, that's really interesting. One of the things that I believe you were discussing in your presentation last night was this idea of like finding the locality of biotransformation sites for drug metabolites, and like how that helps with drug metabolism and pharmacokinetic studies. Can you maybe share an example of like how that works?
Yeah. So, when anyone consumes a drug, right, your body metabolizes it. Basically, at the end of metabolism, you start to see different types of structures come off. that are of the original parent structure but have certain modifications. And those certain modifications can be problematic. And that's why you will have like all these scientists really going in to figure out what those structures are, what those metabolites are. Are they going to cause some type of effect? Typically with a mass spec, you're able to, like I said, you have those puzzle pieces. You can look at it and you can figure out, oh, hey, this is a new type of modification that's happening at this site, and that means X or Y or Z. So it's really important to be able to figure out the structures of those metabolites, but also be able to, figure out the levels of those metabolites as well, which the XenoTOF 8600 system can do with the better sensitivity.
Another thing that I was noticing was that you have sort of like automation support for bioanalytics researchers to like help them go through their workflows more effectively. So one of these was this idea of like an automated fragment selection feature, and then another thing that came up was sort of this metrics tracker tool. So like I'm interested in like what are the additional components of the system around the outside of the mass spec itself that sort of like help to automate or improve the workflows of the bioanalytical scientists?
Yeah, so the software side, right, is the software. Basically, our system comes up with SCIEX software. So SCIEX software really tailors all the features that are needed for quant. So that users can acquire the data, manage the data, as well as you do all your integration processing within that software. There's always aspects where there's, a lot of manual upfront work, like the automated fragment selection, right? And that really helps a lot of folks that are doing quantitation, because in a high resolution mass spectrometer setting, you typically get all the fragment ions coming through, right? So basically, you can filter out and figure out which fragment ion gives me the best sensitivity, which fragment ion gives me the best selectivity, or in the case of, the dynamic range, which one gives me the best or the widest dynamic range, right? And a lot of times you have also the capability of summing these fragment ions, because you can also leverage that level of sensitivity through summing. Because of that, there's a lot of manual work where you would go in and try to figure out what works where and what would work for me. We have that feature in SCIEX software that can now automatically, like you would define your criteria. You could say, yeah, this is the information for my peptides. This is sequence, this is the concentration levels or the types. I want to target lower LOQs, and it will basically take the information from your fragment ions as well as the theoretical fragment ions, match it and figure out this is the final results that will cater to the lower LOQ, or this is the final results that will cater to the broader LVR. So that really, minimizes that upfront work. So you don't extensively have to go for your data as much. And it will give you a nice, peak with integration that's already summed with the different fragment ions that you selected. So that's really, exciting for bioanalysis, because it allows you to minimize that data processing stage, right? And then you mentioned metrics tracker. And that's another feature that we have inside. So it's software that allows you to track the performance of your system. But sometimes, things come up. Obviously, any system, things will come up, and it's a little bit harder to track the performance. And metrics tracker does that, where it will allow you to measure or look at the performance of the system, look at the sensitivity of the system. If there's any, significant drops in sensitivity, hey, I need to schedule a cleaning. I need to do X, to get it up and running again. So.
Awesome. So there's like a component that's helping the researcher with their workflow, and then there's also sort of helping like the machine operations side to like understand like the status of the machine and make sure that they're staying on top of their maintenance, making sure that the machine is operating to the highest degree of quality that it can. For a CRO or another biotech or pharma organization with a bioanalytical workflow that and they're looking at the XenoTOF 8600 relative to the other mass spec machines that they might get, how would you recommend they think about distinguishing like the 8600 from other options that they might get? How would you encourage them to think about like the ROI or the cost of ownership of a machine like this?
So the XenoTOF 8600 system comes with a lot of great features that cater to bioanalysis, right? It is a box that can do both qualitative and quantitative work. We've, added, as I spoke about the XenoTrap and the duty cycle, we discussed that. So it really helps with making sure you have the most sensitive system there for bioanalysis work, where you're looking at some lower level abundance target analytes in your very complex matrix, as well as being able to do qualitative work on the system as well. So the system also has two different types of fragmentation, right? So it has both EAD, electron activated dissociation, and CID, collision induced dissociation, both of which help with identification workflows as well. So if there's labs that are doing both qualitative and quantitative work, it's a really nice, useful platform to be able to have that flexibility in your lab. Both sides of the spectrum, right? Qualitative and quantitative workflows, the XenoTOF is able to cater to that. And the 8600 is very much so on par in terms of quantitative performance with nominal mass spectrometers. So if there are assays, like you saw last night, some of the analytes, so some of the examples that I'd shown last night, right, they had high background issues. Some of them had closely eluding matrix peaks. All those assays can be easily moved on to a high resolution mass spectrometer, not having to do additional sample prep that you would traditionally do with, a nominal mass spectrometer. All of those are very advantageous features of the XenoTOF 8600 system.
So as we come to a close, like just looking at, looking toward the future a little bit, do you expect machines like the 8600 to eventually displace your triple quads like the 7600, or do you expect it to, sort of be used in a complementary fashion, on a forward-looking basis?
I would say complementary.
Okay.
So definitely the triple quad is your gold standard for a reason, right? It gives you very sensitive quant. There are cases where there's complexity. Let's say you want to quantify a structure that's very hard to break down. You want to use MS1 level quant. High resolution will give you very accurate MS1 level quant. Or if you have those background issues, as I had mentioned earlier, you can have more selective and sensitive quant on high res, or at least the latest gen high res, right? I think that they work hand in hand. There's going to be areas where a high res will make sense, and there's going to be areas where a triple quad is going to make more sense. But they both offer good advantages for bioanalysis.
Anytime we talk about data, we end up talking about machine learning and AI. I'm just interested in, what are the intersections of machine learning and AI with either the method development that might go into, how you utilize the machine, or the consumption of the data that's coming off of the machine that you think are sort of like the most interesting applications that can be complementary to a machine like the 8600?
Yeah, I think greatly from the data processing side of things, AI would be really helpful because you get a lot of data, right? At the end of the day, that's also another bottleneck, is how do I reduce my data to meaningful results, like conclusions at the end of the day. So that's really critical. And I think in that area, you would probably need a AI integration in certain places where you can, take a large sample set of data and be able to get the conclusions that you need for your samples.
Have you seen anything in existence that's been moving in this direction, or is it still a little ways out for the ability to do that?
We have AI quantitation software.
Uh-huh.
It's a collaboration with Mass Analytica. We have some examples out there where we used it for a large sample set of data, being able to reduce it down easily, integrate automatically. You will have your user define criteria for what you want to set it up as, what you would like to see in the final results. And then the software will do the magic for you. So yeah, it's definitely out there. And I think it's going to continually evolve as well, because more samples, you're going to get more and more data. And for sure, that's going to be a big area for AI.
even more generally, what excites you about the future of mass spectrometry? Are there other innovations coming down the line that you think are, really exciting or might change things?
Yeah, of course. There's going to be a lot of innovation. As always, continually we're innovating to become most sensitive, being able to measure, across a wide range of concentrations, have these like different types of capabilities, right? Such as EAD, we have a different types of fragmentation mechanism. We have all these new modalities that are coming out, and being able to target all of them is going to be critical for our customers. And so we're continually getting information from our customers, talking to them, figuring out like what are the main challenges currently in the market space so we can cater our next innovation to answer those questions.
What are the most interesting types of feedback that you get from customers and potential customers that sort of influence how you all think about R&D?
Yeah, I mean, we get feedback for our software. We get feedback for our mass spec. How can it be more robust? Last year we had launched our SCIEX 7500 Plus system, listening to customer feedback, understanding that robustness needs to be a critical player. So they have more instrument uptime. They're spending more time doing analysis, right? And we listened and we delivered. We have this MassGuard technology in there to really push for more instrument uptime. And we also implemented it on the 8600 system. So feedback like that really helps us innovate better for the next round of instruments.
Yeah, Eshani, you've been a great guest today. I really appreciate the time. Before I let you go, would you mind telling people where they can learn more about your work and the work at SCIEX?
Yeah, absolutely. You can go to SCIEX.com. We have a lot of our applications there. I work for a global technical marketing team. We have different folks that take care of different market verticals, and we are always continually driving applications with customers, innovative applications. So you can read more about our technical notes there in the pharma, biopharma space, as well as other spaces. And also you can learn more about our products as well. So we have the product pages there too.
Fantastic. Well, Eshani, thank you again. Really appreciate you coming down today.
Thank you. Thank you for having me.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.